Okay, so let's continue the story. I reached the point where we had excitation, contraction, coupling, which means the action potential that traveled along the sarcolemma went down the T-tubule. I was describing it as a roller coaster ride. And this depolarization triggered the opening of voltage-sensitive calcium channels that are in the membrane of the sarcoplasmic reticulum, which is a reservoir of calcium ions. They're stuffed in there. And when these channels open, they're going to come out and flood the cytoplasm inside the cell. And meaning that we have a lot of calcium that's going to be available to the calcium binding protein called troponin. Troponin and tropomyosin are the regulatory protein that are going to regulate if there is going to be contraction or no contraction. So here we are basically activating this troponin with the presence of the calcium. So let's continue the story and discuss what happens at the level of those filaments, those myofilaments. So we're going to have here the let's see, we're going to have thick filaments of myosin, and remember, they are heads, myosin heads that are kind of poking at all of these are thick filaments all throughout, so I'm not drawing every single one of them, but these are all the heads, and they kind of like form the cylinder, and all the heads are sticking on the outside of that cylinder like that. So crisscrossing through this are the thin filaments of actin. The thin filaments of actin are in between, just like that, and there's only one particular region where there's an overlap. Oops, it should be more parallel than that. There we go. So there's a region of overlap over here, but there's also a region where it's only thick filaments and a region where it's only thin filaments. And right where in the middle section of the thin filaments, this would be the edge of the sarcomere. Okay, and you would have more thick filaments down here. So there is, so the pattern repeats itself. I just want to make sure that you realize that there is another sarcomere down here with more heads, etc. So I'm not going to redraw it, but it repeats itself. So you have this pattern of thick filaments, thin filaments, thick filaments, thin filaments, thick filaments, and that's how you end up with this zebra pattern, this striation inside the cell, and you see it under the microscope. Oh, the sunset is amazing. Okay. So that's what I want to zoom in on. I want to zoom in on this section of overlap. All right. So this section of overlap, I'm going to draw it down here, and I'm going to show. I'm going to draw. Now I'm going to zoom in on the actin filaments, and I'm going to draw only 4 globular heads. So if you remember from my other video where I explain actin and myosin filaments, actin filaments are actually, so the F-actin, the filament, is actually made of globular actin, G-actin, so tiny little beads all together next to each other make up the F-actin. And these individual beads actually have a binding site for the heads of the myosin. So this is the active binding site. Normally, this binding site, I'll draw it real quick here. Normally, this binding site has a has a regulatory protein sitting on top. This is tropomyosin, and I think I used this color for troponin, and here's troponin. So troponin and tropomyosin are the regulatory protein, and they're covering that active binding site. So that binding site is actually hidden by troponin and tropomyosin. And maybe we can make this actually, let's do this. Let's make tropomyosin a really thick, a thick molecule, and this is troponin. So troponin is the one that binds the calcium, and when calcium binds, this whole complex moves out of the way. So when this complex moves out of the way, so this is when you have calcium, so in the presence of calcium, you have the binding sites that get revealed because the complex moves out of the way. So when these binding sites are not hidden anymore by those regulatory proteins, the tropomyosin and troponin, this complex that normally hides those active sites, those binding sites, then you have them revealed. So these sites are revealed here, which means they are now available for the heads of the myosins. So the myosins heads are going to literally click in there, and I'm just going to draw two because I want to really simplify this drawing. So you see these heads that are basically facing the actin filaments, these heads. If the complex is out of the way, that means the heads can actually attach to the binding site. So the heads attach to the binding site, and that's the first thing that happens. So there is attachment. So sites revealed. And that means heads attach. So the heads attach, and the next step is there's a ratcheting movement where the heads, we describe them as being in a cocked position when they're attached, and then they flex and pull on the filament as they're flexing. So so then there's a flexing movement. So at that point, these heads, they kind of flex down like that, and this brings the filament forward. So now we're bringing the filament forward, well, forward in this direction. And I'm going to draw this whole I'm going to draw the same for globular actins, but moved by one down. You see how this moved by one down? It moved forward because the heads have flexed. And here I'm going to show the flexed head. There's one and two. So the head was cocked, and then it flexes. And when it flexes, it ratchets, so it moves the filament inwards. So inwards, because this is happening, by the way, this is happening at both ends of the sarcomere, well, at both ends of this A band. This is called the A band. And we're basically having those thin filaments being brought inwards. So these filaments are going to go inwards. And it's called a sliding filament theory because they are sliding, the thin filaments are sliding over the thick filaments. And that's why we call that the sliding filament theory. The thick filaments stay stationary, but with their heads, They ratchet the thin filaments over and bring the muscle, it squeezes the muscle inwards and creates this contraction of the muscle. So of course the muscle is going to shorten in length and pull on whatever it was attached to, and that's how we pull, for example, bones. The next step is very important because the next step is the step that requires energy, ATP. So following this flexing movement, let's add here in our story, we have the filament that slides. Filament slides, but then we have to have heads get recocked, and so to get ready for the next binding. So this is what happens to the heads. So they are in, oops, let's keep the color like we're supposed to. So these heads, they were in this flexed position, but we want them in the extended position, or we also call it cocked position. So for that, to go from here to here, this actually requires an ATP molecule. ATP. ATP comes from cellular respiration. It's an aerobic event. So you breathe in. So when you're breathing in and you're able to make ATP, and this is part of where ATP is going to be consumed to create to extend these heads so they can bind to the next open space and ratchet even more. And that's how we have the sliding of the filaments over and over and over until we reached a maximum point. And that's what happens during the muscle contraction part. So I'm going to write here. So this was contraction, the contraction phase. This is the contraction phase. Okay, and it's very important that you realize there's ATP usage here. Okay. Okay, so relaxation. So how does the muscle relax? The muscle relax in several ways. First, we can have the nerve impulse stop. Nerve impulse stops, which means there is no more action potential. So the voltage gated channels are going to close. The channels are going to close. So there is no more escape of calcium ions, and calcium is going to be pumped back in the sarcoplasmic reticulum. Calcium pumped back in the SR. This requires ATP. So pumped means there is ATP needed. Okay. Also, when there is no more calcium, that means that a consequence of that means that there is the troponin, tropomyosin, so I'm going to write T and T. Complex covers actin. That means the heads of myosins can't bind. And if they can't bind, that means there is no more sliding filament theory, there's no more the sliding filament process, and there is no more contraction, so the muscle relaxes. So all of these have to happen, and this basically gets ready for the next event. The calciums are pumped back inside the SR to get ready for the next stimulation, so they can be released again in the sarcoplasm of the myofiber for the next contraction. So that was the relaxation phase.